I'm Morgan Bohrer.
I'm Michael Akir.
I'm Alex Verne.
I'm Stephen Hahn, and we are the co-founders of TrachTech.
Our design concept was inspired by complications that can arise following endotracheal intubation, specifically those cases involving ventilation. Patients that require mechanical ventilation are often intubated with endotracheal tubes, which can experience a buildup of biofilm, which can then have serious implications. This biofilm buildup directly leads to 80% of antibiotic-resistant infections, the most common being VAP, or Ventilator Associated Pneumonia. VAP is a lung infection that develops 48 hours or longer after mechanical ventilation has begun. This form of pneumonia occurs in 12 to 25% of all intubated patients, and is fatal in 24 to 71% of those cases. Biofilm buildup will also increase the airflow resistance by decreasing the diameter of the ET tube, causing spontaneous breathing tests to be more difficult for the patients to pass. This medical complication is serious given any circumstance, but in the wake of the COVID-19 pandemic, the impact of ventilator-associated complications is unprecedented. In the past few months of 2020, the production growth of ventilators has almost doubled. It is common for a tracheotomy to occur after a few days of a patient being intubated. Now, with the highly infectious nature of COVID-19, tracheotomies are being postponed for as long as possible to limit exposure to the virus. With ETTs being left in the patients for longer, there is a greater biofilm buildup that will occur, which only increases the chance of developing VAP and other serious infections. The time for a viral clearance is also increased in patients that are intubated, and these patients are typically testing positive for COVID-19 for longer.
Given the severity of the issue, we decided to take on the challenge that medical professionals need a way to remove built-up bacteria or biofilm in patients that require endotracheal intubation in order to diminish adverse effects without reducing the efficiency of oxygen delivery. After many hours of brainstorming and rapid prototyping, we finalized a design that best met our criteria. It featured three main components: a helical tip, catheter tube, and driver motor. The helical tip is the most important aspect of the design and is specifically designed to remove biofilm from the inner walls of the tube while maintaining airflow and not moving the endotracheal tube or applying pressure to the trachea. As the tip spins, the helical flutes sweep the inside of the tubes, shearing biofilms off the walls. The angle and pitch of the fins move the debris up and away, preventing it from falling into the lungs and keeping the tip clear to continue cleaning. The rotational motion of the tip will not exert forces outward onto the endotracheal tube and prevent soft tissue damage and ulceration of the trachea. The center of the tip is ported to allow for airflow from the ventilator to flow through the catheter, out the tip, and into the lungs. The catheter also acts as a drive shaft, translating rotational motion of the motor into the tip. The material of the tip is a medical-grade silicone rubber that is stiff enough to exert shear forces required to remove the biofilm, but compliant enough to not damage the endotracheal tube. At the moment, however, we have been using a hard resin 3D printed plastic to create and test designs. The size of the tips will be created to specifically match the common endotracheal tube sizes, averaging between 7 and 9 millimeters with a half millimeter step in between. It's important that each tip be fit to the endotracheal tube so that the flutes of the tip can effectively sweep the walls and remove biofilms.
Our design is similar to a product on the market currently called EndoClear.
EndoClear is also intended for removal of mucus and bacteria secretions from the inside of endotracheal tubes. This product differs from ours because they rely on a balloon that is used to wipe away loose biofilm on the inner surface of the tube, while our design uses spiral flutes that produce a rotational motion to dislodge the tough secretions. We attempted to buy samples of the EndoClear device so that we could do side-by-side testing against our prototype, but we were unable to purchase it from the company.
The first test we conducted was a mass removal test using sections of soft PVC tubing filled with petroleum jelly. The testing rig was 3D printed in the makerspace to hold the tube steady while the device was passed through. We conducted 5 trials and had an average percent removal of 53.04%. After reaching out to the Tulane Simulation Center, we realized that petroleum jelly was not an accurate representation of biofilm buildup, so we decided to use uncured silicone for our tests. We also used an improved test rig that was water jet cut from aluminum to improve stability. 3 load cells were attached to the middle of the test rig. These were used to quantify the force exerted by the device on the tubes as the device was passed through. The load cells measured the forces exerted in the X, Y, and Z directions. Finally, we wanted to compare our various design iterations, so we performed tests on tip 3, tip 4, and tip 5. 10mm diameter soft PVC tubing was cut into 6-inch sections in order to fit the new test rig. These sections of tubing would represent a section of an endotracheal tube. Each of the tubes was weighed in order to determine percent removal later. The blue liquid silicone was poured into the tubes until it covered the entire length of tubing. The tubes were then held up so that any silicone not sticking to the edges would fall out. Finally, the tube was weighed once again to determine the filled weight. After being filled with silicone, the tubes were inserted into the testing rig. The device was turned on and passed through the section of tubing, which took about 8 seconds on average. The cleaned tube was then weighed. The clean weight, the filled weight, and the empty weight were used to determine the percentage of removal. This number was used to evaluate the effectiveness of the device at mass removal. Tip 3 snapped in half a few inches inside the tube on its first trial. Since we only had one printed version of Tip 3, we were unable to test it further. Tip 4 had a removal percentage of 50% over 5 trials. Tip 5 had a removal percentage of 92%. It was by far the most effective design in terms of mass removal. Tip 5 also experienced a lateral force of 136 millinewtons, compared to 313 millinewtons for Tip 4, showing that Tip 5 would put significantly less force on an endotracheal tube inside a patient.
Looking forward, we plan to expand on our current testing protocols. We hope to conduct more tests that will show us the most efficient way of removing biofilm.
We plan to test if the speed of the motor will impact the removal of biofilm, in addition to testing variations of the tip length. We have received funding from Schlumberger to purchase the EndoClear device, but due to the COVID-19 pandemic, we were unable to get it in time. In the future, we hope to get in touch with EndoClear and complete our comparison testing. We are currently well under budget for our device.
We are currently in communication with a patent attorney to see if our technology can be patented. We are in talks with the teaching team about allocating part of our budget towards our patent process since our device was relatively low cost.
For a more in-depth analysis of our design, feel free to visit our website at trachtech.wixsite.com/TrachTech. Here you can learn more about our team goals and why we decided that biofilm buildup was an important medical issue that we wanted to tackle, especially in the wake of COVID-19. You can also find additional images and descriptions of our device, as well as testing results. We have also included images of our time spent creating this device over the course of the year. Finally, all of the TrachTech organizers would like to take the time to sincerely thank the Schlumberger Foundation, our community partners, the First Robotics Competition, and of course, the Tulane Biomedical Engineering Department. None of this could have been accomplished without the help of Dr. Catherine Raymond, Dr. Mark Mondrinos, Dr. Chrissy Guidry, Terry Ferret, and our TAs, Scott and Afshin. Please feel free to contact us with any inquiries below. Thank you so much for your time.